Hi everyone, welcome to iVoM. I'm Joana Azeredo from the University of Minho, Portugal, and together with Madalena Pimentel from the University of Lisbon, we are the organizers of the Virus of Microbes 2020, which will be held in Portugal. As most of you are aware, due to the COVID-19 crisis, the Virus of Microbes 2020 conference is postponed to July 2021. So, to fill in this temporal gap and to stimulate and keep connections among the community working on Virus of Microbes, the VOM 2020 Organizing Committee, with the support of iSVM and phage Directory, are promoting iVoM under the theme Take a Walk on the VOM site.
Hi, I'm Madalena. For the iVoM, we'll have 6 webinars, 1 per month, each dedicated to each topic of the VOM 2020. The first one starting today and ending on next February. On behalf of the organizing committee, we would like to thank all the speakers, sharers, sponsors, and the audience for helping us making this possible. We welcome all to this first iVoM webinar, and we hope to see you in Portugal next year. We leave you now with the 2 chairs of this session, Corina Brussaard and Debbie Lindell. Corina, please introduce yourself and start this section.
I'm Corina Brussaard. I'm the president of the iSVM, and it's really my honor that we can actually open this first session here. Together with Debbie Lindell, we will be your chairs. We have 3 speakers for today, and the topics are based on distribution, diversity, and evolution in that order. And 2 of the talks, number 1 and 3, deal with the fascinating story of the Crass phages. The second talk focuses on the viruses of phytoplankton. I would say let's start with introducing Rob because of time. Let's just press on. The first speaker is Rob Edwards, and he is recently migrated from San Diego State University to Flinders University in Australia. Rob is a fellow of the American Academy of Microbiology. He's one of the pioneers in the Crass phage story, so the human virus side. And beside that, he also works on coral viruses, so he's pretty broad. And I'm really looking forward to your presentation, Rob. So the floor is yours. The screen is yours.
Thank you very much. And I'd like to thank the organizers for inviting me to talk and for actually accommodating me as well, because here in Australia, it's about 8:30 in the evening. And so I sincerely appreciate you guys not making me stay up until 2 a.m. to talk. As Corina said, I've just moved from San Diego to Flinders University in Adelaide. And at Flinders University, we like to acknowledge the traditional owners and carers of the land upon which the university resides and where I live now. And so we're thankful for their support. And what I'm going to tell you about today is how we think the Crass phage is spreading both globally, regionally, and locally, and the differences between those. Before I just tell you that, I'm just going to tell you one thing about what really attracted me to Flinders. I'm now the Flinders Fellow in Bioinformatics, but I'm actually the director of FAME. The Flinders Accelerator for Microbiome Exploration is definitely a backronym. The dean of the college wanted fame and fortune, so we bought FAME, and hopefully he'll bring us fortune. And FAME is a new initiative that we've launched really across South Australia, where Adelaide is, and Australia as well, to support metagenomic and microbiome research across the whole gamut of things. We're hiring postdocs, we're recruiting people to work with us, and so if you want more information about that, just direct message me on Twitter or send me an email or get a hold of me somehow, and I'd be happy to talk some more about it. But let's talk about Crass phage. Crass phage was a virus that we discovered. We published it in 2014. It was discovered by Bas Dutil, who's now an assistant professor at University of Utrecht in Holland. And he was looking at a lot of metagenomes from, in particular viromes, from gut samples from a twin study done by Forrest Rower and Jeff Gordon in St. Louis. And Baz identified something a little unusual, which was that there's approximately 100 kb phage genome that was present in all of the twin studies that we looked at, and there are about a dozen or so samples in that study. At that time, we expanded it to look at all of the metagenomes, especially human-associated metagenomes. Luckily, in 2014, there weren't nearly as many metagenomes as there are nowadays. And we showed that it was present in about half of the human metagenomes. And that was a pretty striking finding at that time. And so Baz and I like to go around the world and give talks like this and tell people what we've been finding. And everybody says, you know, you keep saying it's so important, go prove it. And so we, being in San Diego at the time, here's San Diego State University is up here where the little icon is. This is the Bay of San Diego. And San Diego is right on the southern part of the United States. And this line is the demarcation of the border with Mexico. And so we decided to go and see if we could identify Crass phage. And of course, what better place to go to find something so good as to go to Mexico? And it turns out that there's a sewage plant down here on the border that takes the sewage from Tijuana and treats it so that the sewage does not flow out into the ocean and up towards San Diego here. So we went down to the sewage plant. We asked them for a little bit of influent. On our first visit, they tried to give us 60 liters. We only really needed a few mil. And in fact, we only really needed a few microliters because from about 2 microliters of influent, we can PCR Crass phage. So we use these samples to optimize the PCR reactions, the DNA isolations, and we sequenced a lot of Crass phage from around San Diego. So we had this sort of crazy idea to just email people and say, hey, we've got some sequences, some primer sequences, and you can probably get some sewage. How about you go and get some samples and do the PCR and send us the sequence, and we promise we'll add you to a paper. Fortunately for us, quite a few people believed us, and we ended up with Crass phage sequences from about 2/3 of the countries in the world. So the map here shows the distribution of samples that we got. The blue dots are where we only had one sample, one sequence from a site. And so we can't tell anything about the global spread of that virus if we've only got one sequence. The larger circles show where we have more than one sequence per site. And the red is the proportion of sequences from that site that are most similar to other sequences from the same site. In contrast, the yellow is the proportion of sequences from that site that are most similar to sequences from other places, not the same site. And you can see for the majority of places that we've looked at, most of the sequences are more similar to sequences from where they came. We can actually go a little bit further, of course, because we've got the sequence of a fragment of the genome, and we can build a phylogeny. So this is a phylogeny of several thousand genomes that we have isolated from Crass phage. And all of this data is on GitHub. You can go download it. You can repeat the analysis. You can add your own Crass phages, and you can see where they lie. But the real take home from this was that if you give me a Crass phage, I can tell you the country that it comes from with quite a high level of certainty. One of the things we don't know yet is how long it takes Crass phage to ameliorate. And, you know, perhaps if you just move from, let's say, the US to, let's say, Australia, maybe you're thinking about how you could study that. So that's at the country level. So then we went down to a slightly smaller scale level, and this work was done by Brussaard from the University of Warsaw, who I saw managed to sign on to this. So, hey, Brussaard, it's good to see you again, almost. And Brussaard got Crass phage samples from several different cities around Poland. And we did the same thing. The table here on the right shows the distance between the cities in kilometers. And the phylogenetic tree has been colored by the city that the Crass phage was isolated from. And you can see that at this range, at this scale of about 100 to 200 kilometers per sampling site, there's still a very strong regional signal. So you can go and sample somebody's Crass phage and not only say what country they're from, but what city they're from. And so then we went down a step further and repeated this work in San Diego. And down the bottom here is the original Tijuana sample that I showed you. We have 4 other sites that we sample regularly up in northern San Diego County, heading up Los Angeles is just off the map here. And these sites are much closer to each other. And so they're less than 20 kilometers apart from each other. And Alex Vega, who's shown here, collected a lot of samples over time from these sites. And what we see in these samples is not that strong geographic signal, but rather more of a temporal signal where our sequences now cluster by the date of isolation. Of course, we've all been affected by another virus, and I can't really let talk about viruses pass anymore without mentioning COVID. And these figures I just took from NextStrain this afternoon. The figure on the right shows the number of genomes, it's nothing to do with the incidence of disease because the US is so small. I can say that now, I don't live there. And the graph on the left is colored by the country of isolation. And so, of course, we know that COVID is evolving as it spreads around the country. And then I just had to show this figure because, well, basically we don't have COVID in Adelaide. And so after this, I'm going to go to the pub without a mask and have a good time. But basically, even in small regional areas like in the US, in Australia, where we've had outbreaks, there's a very strong regional signal. And so I think there's a few outstanding questions about how the phages are spreading, especially in this situation where we have a tripartite relationship between the phage, the bacteria the phage infects, and the human host as well. And we're also really curious about both the temporal and geographic variation of Crass phage. Is it really just a kill the winner, where a bacteria grows, it becomes resistant to a phage, so it can grow up, there's a mutation in the phage that now makes it able to kill the host, or is there other kinds of selection at play in this? Is there some seasonality in the data? These are all great questions that we're trying to answer. As I mentioned, there was 115 people on the Crass phage paper, including many, many people that I'm delighted to see on this conference. And we could not have done the work without each and every one of them providing DNA sequences, and of course without Baz. And I'd like to finish by wishing everybody happy International Microorganisms Day. Thank you very much.
Thank you, Rob. Thank you very much for that introductory talk for this webinar series. Every participant could hear the gong. That is 2 minutes before the end, and the end, just that you know that you recognize the sound. I'm giving the floor to Debbie, who's going to introduce the second speaker.
Okay, hello everyone. It's so nice to see so many participants. My name is Debbie Lindell and I'm from the Technion Israel Institute of Technology. Before I introduce our next speaker, I just wanted to mention that if you have questions to Rob and in fact to the other 2 speakers, you can write them in the chat and questions will be asked at the end of the session. So it is now my great pleasure to introduce Ruthanne Sander, who is a professor of biology at the University of Bergen in Norway. Her research focuses on virus-host interactions in marine algae with the aim of gaining a general understanding of how viruses affect the biodiversity of their host populations and thus how they affect the marine microbial food web. Today, Ruth-Anne will speak to us about the adaptive evolution of haptified infecting viruses from those that cause acute infections to those that stably coexist with their hosts. And as you can see, the title of her talk is Diversity in Marine Algal Virus Host Relationships and Their Effect on Phytoplankton Community Structure. Take it away, Ruth-Anne.
So, yes, now I'm finished. So, Okay, so the diversity in algae viral host relationships in the ocean is huge, and I will in this talk come up with some suggestions on basic factors that might be crucial for the regulation of these relationships by focusing on a key phytoplankton group, haptifieds, and their giant viruses. According to evolutionary theory, every new viral host relationship starts with an acute phase where the virus is highly virulent, having short infection time, and giving rise to high host mortality. However, the virus-host relationship will evolve as the two players adjust to their new symbiosis. The virus has to balance its virulence within the host with, for example, traits like transmission to other hosts, resulting in development of less virulent viruses. The host, on the other hand, has to balance its defense against the virus with trade-offs such as, for example, reduce competitiveness for uptake of limiting nutrients. And some traits that can be used to describe algae viral host relationships, as it will affect the viral fitness, are the latent period of the virus, that is, how long time it takes for the virus cell to produce viral particles, decay rate of the virus particles in the environment, the host range of the virus, the virulence of the virus, that is, the degree of host mortality the viruses cause, infectivity, that is the percentage of infectious particles of total particles produced in a lytic event, and the mode of transmission of the virus if the virus is transmitted horizontally or vertically to their hosts. And further, host traits of importance for the relationship will be, among others, the growth strategy of the host, if it is an R or a K strategist, and the host resistant mechanism against the viral attack. And again, all these fitness traits will come with a biological trade-off. So, can we use this information about viral host relationships and traits to better understand viral host systems in the ocean? In the marine environment, we have many examples of boom-burst relationship where a specific virus can lyse a dense bloom of R strategist hosts within hours. And this resembled more an acute lytic relationship with highly virulent viruses and short duration of infection or short latent periods. However, seen from metagenome data, there are many examples showing a dominance of specific viral OTUs that are present at several time points during long time periods, as shown from these two experiments that were conducted over 1 and 2 years period, where, for example, the green or the light blue viral OTUs were present at nearly all sampling points, and such pattern indicate a more persistent relationship with the host. But little is known about these systems here, as they are difficult to culture due to their low abundances in the ocean. So haptifieds comprise a diverse group of marine algae, dominant and widespread in the ocean, consisting both of bloom-forming R strategist and non-bloom-forming K strategist. The haptified viruses isolated so far have double-stranded DNA genomes and belong to the nucleocytoplasmic large DNA virus group, the NCLDV group. Most of the so far isolated haptified viruses belong to the Mimiviridae group, while also some belong to the Ficovnaviridae. And recent studies have shown that these haptified viruses are very abundant in various oceans, as are their hosts. Those haptified virus systems in culture show a continuum of infection stages from acute infection, rapidly lysing their host, to more persistent infection with lower host mortality rates. And we believe that this range in infection strategies among haptified viruses has evolved as a response to the range of haptified growth strategies. So the scarce data and available on fitness traits and trade-offs in a haptified viral-host relationship show that there is a tendency of those viruses infecting bloom-forming hosts, our strategists, to have shorter latent period and also also higher infectivity, as shown here, resulting in higher host cell death compared to viruses infecting K specialists. Also, these are specialists infecting specific strains within a species. And these are traits that are characteristics of an acute relationship. Virus infecting K strategists, on the other hand, have a tendency of having longer latent periods and being less virulent, resulting in lower host cell death compared to the more acute system. And here there are several isolates that are generalists, that is, able to infect different species. And all these traits fits with characteristics of a more persistent relationship between virus and host. So our hypothesis is that the host growth strategy have a major effect on the development of different viral infection strategies. The diversity in infection strategies for haptifieds and their viruses span from acute system with viruses infecting bloom-forming haptifieds with our strategy to more persistent systems where the virus and host are co-occurring year-round at low abundances together with other microalgae virus systems with K strategists as host. And we suggest that these different infection strategies come with differences in fitness traits and biological trade-offs. The acute systems are characterized by short latent period, highly virulent viruses with high infectivity and decay, narrow host range, and as their hosts are abundant, they are transmitted horizontally to their host cells. We also suggest that these viral host systems have a higher diversity on the strain clone level due to the higher host growth rates. And this can be explained by the virus lysing the most fast-growing host, enabling coexistence between slow and fast-growing host populations. We further suggest that the most common viral host system in the ocean are those of a more persistent nature, consisting of low virulent viruses, employing a variety of strategies to coexist with their K strategist hosts. And these systems are characterized by viruses with longer latent period, lower virulence, infectivity, and decay compared to the acute system, and might have broader host range. Long-lasting infections, as hosts become scarce, will increase the chance of vertical transmission rather than horizontal transmission. And these systems here might often be neglected due to their low abundances, but as they are most common in the ocean, they do have important ecological roles. So these are questions that we are working with in the Virby project, where we try to answer these hypotheses by using viruses having different infection properties and their hosts as model systems in controlled experiments to dissect their infection behavior to better understand viral ecology and evolution in the ocean. So thank you.
Thank you very much, Ruth-Anne. That was fantastic. And I will now transfer this over to Corina to introduce our last speaker of today's webinar.
Yes, thank you, Debbie. Yeah, the last speaker of today already is, it goes fast when you're enjoying yourself, is Colin Hill. He's from the University College in Cork in Ireland. And as we now heard from Rob, we have, most of us have this Crass phage in our gut, right? But we don't really know the host. There was only the silicon predictions. And Colin has actually been able to isolate the host. And his talk will be about the co-evolution of the virus with the human gut microbiome. So I'm giving the floor to Colin. And again, sorry, Colin, sorry to interrupt. I'm not giving the floor yet, sorry. just to make sure people just write your questions in the chat and we will discuss them after Colin's talk. So now the floor is yours, Colin. Thank you.
Okay, thank you very much, Corinne. And I hope people can see the screen okay. And so what I'm going to do is I'm going to just continue the story that Rob already started for us on Crossstage. And so luckily, well, just so I will move on. Okay, so luckily, I anticipated that Rob would have already introduced Crossstage, but here's the Crossstage CV again in short. It's the most abundant phage in the human gut. It's co-evolved with the human lineage, and of course we owe a great debt to Rob and Baz for identifying this phage in silico in the first place and showing us the global distribution for that phage. We became interested in following that paper, of course, in this extremely abundant phage. And what we started doing was assembling phages from different individuals. And we came up with a Crossstage taxonomy in 2018. This was work done by Andrei Shkoparov, Emma Guerin, and Steve Stockdale in the lab. And in that, we characterized one family, 4 subfamilies, 10 genera. And this already seemed complicated enough. But in fact, we've now just submitted a proposal to ICTV with a whole panel of people where we suggest that Crass are actually an order of phage containing up to 78 different genera. So the situation is only going to get more complicated. And what's strange about these phage, I guess, is that 2 phage can both be Crass phage, but share almost no DNA sequence identity. And that makes them very difficult to study. But here's an example of one genome from each of the 10 genera that we identified in 2018. And you can see that they do have some consistencies. The genome sizes range from about 90 to 105 kb. They're transcribed in 2 significant domains, and there's clear genomic symphony. You can see that the genes are organized in a very similar manner, particularly a very large RNA polymerase down around the 6 to 7 o'clock region there of those phages. But if you look at 7 and 8, Crass phage 7 and 8, you see that they're littered with tiny open reading frames, not showing the same symphony. But this was resolved when we were able to identify that genera 7 and 8 actually use an alternative genetic code. And when you use the alternative genetic code, then the genes become obvious. And again, we can see the same level of symphony. But I think it's amazing that within the same family of phage, you get different genetic codes as well as this very low level of DNA identity. And one other thing that was kind of curious here was that we were able to identify 2 of these Crass phage in the gut metagenome of Utsi, who's lived 5,000 years ago in the Alps. And so this phage has been with us for at least 5,000 years, if not even longer. And so we started studying the gut virome of individuals and looking at it over time. So what the panels here show are each one of these, each number is in a different individual. And we looked at their gut phagome at 12 time points, obviously 1 month apart. And hopefully you can see that people like subject 918 and subject 924 are dominated by Crass phage. This is the purple phage that you see here. And in fact, in some individuals, Crass phage can be 95% of all the phage in their gut at a particular time point at a particular month. And it can represent up to 20% of all the DNA you recover from the feces of an individual. So this gives us a sense of just how abundant this phage is in some people's microbiomes. Now, some people appear to have very little Crass phage, but that's just because of the magnification here. Everybody, in fact, has Crass phage that we've looked at so far. And this just illustrates this. If you look on the left here, you see qPCR of Crass phage abundance, one particular Crass phage in those 10 individuals across all 12 time points. And at every time point in every individual, we can identify Crass phage. Some people seem to be persisting high carriers of Crass phage, and some people low carriers, but everybody has it at all time points. And to even further complicate that, if you go down to this bottom left-hand side, you can see that some individuals like 918, 924, 923 carry a wide variety of Crass phage at any given time. So it can carry up to 7 or 8 different Crass phage at all time points throughout the year. And here's one individual, subject 924, carrying 7 different Crass phage in various different ratios to one another, but all consistently maintained over the year. And if we look at individual Crass phages within an individual, then we can see that even within a given Crass phage, SNPs appear and become dominant over that year. So this is the months of the year, 1 through 12. You see that some SNPs appear and become dominant, other SNPs appear and then disappear again. For some Crass-like phage, there are very few SNPs appear over the course of the year. So even within us, of course, as you might expect, the Crass phage are evolving from month to month, and different SNPs and different clonal varieties are appearing. So a very complex setup. Some people have high Crass, some people have low, some people have multiple Crass phage, some people have very relatively few, and the Crass phage that we have are mutating and evolving all of the time. What we really wanted to do was kind of plant a flag for the wet lab scientists that all this in silico work is fascinating, and the problem with it, of course, is there's never an end to it. You can always do another in silico analysis, but we wanted to actually grow the Crass in the lab. And Andrei Shkaparov, in a huge effort, did manage finally to cultivate the very first Crass phage, which we named Crass 001. And he was able to cultivate this on the organism Bacteroides intestinalis. And this was the one predicted, or Bacteroides at least was already predicted by Baz and Rob to be the host of Crass phage. So this became the first cultured Crass phage, 102 kb genome, and it infects Bacteroides intestinalis, as I said. And we've now gone on to generate 4 other cultured Crass phage in the lab. Second one that was isolated here in Cork by Emma Guerin, and 2 more that were very kindly shared with us by Andrew Hirkoyan and Justin Sonnenberg. They identified it in another study, and all of them so far target Bacteroides species. And of course, being able to grow them in the lab means we can study them. We can look at their plaques, we can look at them under the electron microscope. This is the DNA wrapped in a beautiful kind of crystal structure within the head of a Crass phage. And we can, of course, look at the Crass phage actually replicating within their bacterial host. So we can begin to study their biology and begin to unravel some of these interesting questions regarding how they interact with their host, and of course then in the human host. And the biology has turned out to be extremely interesting. Now I'm only showing some snippets here. If you look at the EMs at the bottom, you can see that 45 minutes after infection, these strange kind of inclusion bodies start to form within the infected cells. Then you can hopefully start to see the phage appear after about 90 minutes. Of course, they build up within the cell over a couple of hours, reach very high numbers, and eventually these cells lyse. And now you can see next generation of infection. Apologies, I moved on a bit too quickly there. You can see the next generation of cells becoming infected by multiple phages after the first burst. But what's interesting is that both in test tubes and in mice, these are mice monocolonized with B. intestinalis, when we add Crass phage, the Crass phage settle into a kind of a relationship with their host. They don't disappear, they don't dominate, they don't wipe out their host, but they live in some kind of coexistence, fairly peaceful coexistence with their host. So both phage and host exist in high levels within both in vitro and in vivo systems. And of course, we can begin to look at the transcriptomics. And here you see that the bacteriophage genes are switched on in 3 very discrete modules over time, while the bacterial genes are all switched off early in the infectious cycle. So a very classic kind of phage infectious cycle there seen at the transcriptome level. It also means that luckily we can begin to study the structure of the phage. And this is something we've been doing with Fred Anson and Ollie Bayfield at the University of York. And I hope this video is working. And you can see this absolutely beautiful Crass phage podovirus morphology. Here's the tail with the channel through which the DNA is ejected. You see this highly sophisticated tail proteins and the phage head proteins. And Fred and Ali have worked this down now to about the 2 angstrom level. So we're really looking at the individual amino acids within the capsid and the tail of this extraordinary phage. So we hope to report this and publish this pretty soon. And so in this very brief talk, what I wanted to do was say that Crossstage so far is turning out to be an extraordinary story, and being able to study it in the lab, of course, is, I hope, really expanding our knowledge of this interesting phage. But basically, they're diverse, they're ubiquitous. We haven't found a human yet that does not have Crossstage. We've now identified over 1,500 individual Crossstage genomes from different individuals. They're highly stable in the gut. They've evolved with the human and animal gut, and they've been with us for a minimum of 5,000 years and almost certainly longer. They can be transferred between individuals. Here's an experiment done by Lorraine Draper in collaboration with Reetta Satakori in Finland. What we showed that when FMT is performed, then if the donor is high in Crass and the recipient is low in Crass, we see a transfer of the donor's Crossstage to the recipient, and then they remain stable over 12 months in the recipient. So you can transfer Crass from one individual to another, and they will engraft in that new host. They coexist with their bacterial targets in high numbers. They're certainly lytic, but they seem to have some kind of a benign interaction with the target, or maybe even a positive interaction with their targets. And so I hope you'll agree that this phage, we still have a long way to go, which is why I only said so far, in understanding the biology and the mechanics of this phage and how it coexists with its host for such long periods in the gut. And I'd like to finish by thanking the people from the group that contributed to this part of the story, at least, Andre, Katja, Steve, Emma, Lorraine, and Paul. And thank you for your attention. Thank you very much.
Thank you, Colin. This is definitely a fascinating story, which we will for sure will be studying for a very long time, because there are some intriguing questions still to be asked there, especially this lytic versus, like having the host still there. Thank you of all speakers for this interesting session. This iVoM, as we call it now. So this is part of the ISVM, the International Society of Viruses of Microorganisms. We will start now with our Q&A, and I have some questions selected from the questions that were directed to Rob. After that, Debbie will take over, and at that point, I will also pop up a slide about the ISVM, and I hope as a true virus of microbes lovers that you are, if you're not a member, that you sign up. It is for free, so it would just be good to have a good group of people that is interested and thinks it's important to get viruses of microbes literally on the map. So first question to Rob. One of the questions was like, at what percentage of sequence identity or the number of changes do you consider a grass phage to be different?
Well, I think that's a great question, and I think Colin just showed beautifully that you can go anywhere on the scale from 0 to 100, right? It seems to be, do you want to be an individual isolate, or do you want to have a completely different genome? And so in our analysis, we've done both, looking at like more strain, trying to identify more strain-specific differences, but then, it goes all the way to completely different things, as we've just seen.
So that means actually that you're not really sure yet.
No.
So it's going to be interesting to look at the taxonomy of the Crass phages then in the future. Another question is, what is the effect of season on the distribution? Because you showed a plot where you say the different month, but do you have, can you speculate what the effect is on the...
Yeah, I mean, I think seasonality is something that's really come to the forefront. I mean, with the COVID stuff, people are talking more and more and more about the spread of viruses, and the general population talking about this in ways that we haven't really considered before, right? And as we're changing seasons, we here are coming into summer, and people in the northern hemisphere are heading into winter. I think people are really worried about the effects of that on COVID. And I think the same is true with Crass phage and with these other viruses. We just don't know what those effects are. And the effects, it really is that tripartite relationship between us and the things that we do, right? So in the summer, we're outside more, we're being more active. In the winter, we tend to be inside more, we tend to be sharing our bugs more. And so it's that interaction between us, our bacteria, and our viruses. And I think each of those things is going to play a part for sure.
So do you think it's a general feature? I mean, you already bring COVID into the discussion, so you think that this is generally happening?
Yeah, because the spread of these human-associated viruses is really dependent on the humans, right? They're dependent on our hosts. We don't have any evidence, any places where we found Crass phage outside of humans, and so the spread that we're seeing has to be mediated by human-to-human interactions, just like it is with other viruses like COVID. And
have you checked whether there is a food, what people eat? Is there a relationship with what people eat?
So we could not find any relationship between what people eat. We used quite a lot of data from some of the big European studies. There's a weak but statistically significant correlation between a high protein or low carb diet and the presence of Crass phage. But we were unable to find any real driver of that. And I don't know whether Colin's found that. Would be a good question to follow up with him on.
There's a follow-up question on the first one, because you stated that if two viruses have 0% identity, then that sounds like a paradox, it's said here. How can you...
Well, I think Colin just showed us beautifully that you can have viruses with basically no genetic, like DNA sequence level identity, but you can identify that very clearly they're from the same family because of the genetic syntety. You know, we know that the viruses undergo much more rapid evolution than other organisms, but they still have to maintain some structure and some order. And so I think, you know, then it comes down a little bit to clumping and splitting, whether you want to call those the same or different. And as Colin mentioned, there's a proposal in front of the International Committee for the Taxonomy of Viruses to consider the nomenclature for all of the Crass-like phages that we've been able to identify so far. So I think that that's a real positive thing that's going to help us sort of set the stage, not just for Crass phage, but I think it's going to be true for other viruses too.
Yeah, I completely agree there. That's the last question because of time. There was somebody wondering, I myself was wondering that as well. Did you take samples, for example, from your own move from the US to Australia to see how long it takes before there is a change in the signature?
So I actually did not sample myself. The main reason that I haven't sampled myself is that I actually spent 28 days in quarantine to get here so that I could go to the pub tonight. So there was a period where it's not going to be quite as straightforward as that. But we've been planning and thinking about some experiments that would help us really try to understand exactly how that change occurs. So that's something we're thinking about.
Thank you very much, Rob. Thank you, the audience, also for the questions. It's really nice to see so many people interested. I'm giving the floor to Debbie for the questions on speaker 2 and 3.
Okay. All right, questions now for Ruth-Anne. First question is, what does the decrease in phage number indicate? Is this the instability of the phages?
I'm a bit uncertain if virus number or about the question. Decrease in, is it decrease in virus number in which incident?
I don't know, it's not my question.
What
does decrease in phage number indicates? Is this the instability of the phages? I don't have more than that.
Okay, well, maybe it has to be linked to the abundance of the viruses. That what I said, that the most abundant ones are those that are more acute systems, while those that are at lower abundances but present all the time or at several time points during a season or during a year are more stable and is also more persistent system or represent more persistent systems. Because I think that's the only thing I have been talking about when it comes to numbers of viruses.
Okay, yes, the person verified that it was the abundances of the phages, yes.
The abundances,
So,
okay. So with acute systems, they will have kind of specific time points during a year where they will have a large bloom of host cells, and this will be followed by a large abundance of virus produced. But for the persistence systems, as I said, they will be more common. They will be present over longer periods, maybe the whole year along, but at lower abundances.
okay, let's go on to the next question.
I hope it answered the question.
Okay, next question. Very interesting, thanks. I have a question about the diversity of viral populations infecting R and K strategists respectively. Do I remember correctly that you said viruses of R strategist by trend show higher diversities? And are there examples that corroborate this trend?
Well,
I'll let you answer that first, and then I'll go into the next part of the question.
Yes, what I said was that those with those acute systems or that infect R strategists, those viruses, they will be creating higher diversity on a clonal or strain level. So it's on this level, this system will create larger diversity compared to persistent systems. So for persistent systems, well, this is it's just a hypothesis, so I can't really say I don't have any examples on this, but what we believe with persistency systems is that then you have also viruses infecting generalistic viruses able to infect different species. So then the diversity will be created on a higher taxonomical level, not on a clonal strain level.
Right, so this relates to the next part of the questions. I was wondering if the blooming of the certain host genotypes of the R strategists could provoke selective sweeps that would reduce virus diversity. So the opposite pattern may also be observed.
Yeah, but what I have seen from metagenomic data is that for these acute systems, you very often have high diversity on quite similar viruses. So clonal viruses that are a bit different, but still you have a high diversity on this level. So I'm not sure if I answered.
Okay, well, if the person wants to add more question, they can. In the meantime, I'll ask the next question, and if the person who asked that question wants to add an additional one, they can. Can you kindly give further clarification on the low infectivity of isolated marine algal virus and its broad host range?
Okay, yeah, what we have seen, we have actually done some experiments with these systems that I was talking about, and some of our viruses that are able to infect two different species, we have shown by NPN, where you can measure the percentage of infection particles, that it's lower, this percentage is lower for those that were able to infect different species compared to another viral host system that we have in culture that is just able to infect one specific strain within a species. And this was measured by NPN. So it's a method where you can actually determine a number of infection particles. And then we compare this with the number of total particles that are produced during the lytic event.
Okay, well, thanks very much. I think I will, we'll ask one more question that's just come up in the chat. Thanks, I think it's clear now. So we would be only higher strain diversity for viruses that infect R strategists. This is just a follow-up.
Yes. Sorry, I didn't get that one.
So it's saying it would be only higher strain diversity for viruses that infect R strategists.
Yeah.
Okay.
Yeah, correct.
All right.
According to the theory. This is just a theory.
Yeah. Okay, terrific. Thank you very much, Ruth. And I'm going to now move on to questions for Colin, for which there are many questions. One question that I have actually is, do you think that this species of bacterium is the only host for the Crass phage, or do you think that it might also infect other bacteria in the gut?
Yeah, so far our evidence would suggest that it's limited to members of the phylum Bacteroidetes, but certainly not just to Bacteroides intestinalis. The 4 phage that we have infect 3 different species of Bacteroides. I think there's a very good chance that Prevotella have Crass phage or Crass-like phages as well, and so certainly much broader than just Bacteroides intestinalis, yes.
Okay, and this might be related to the lack of my knowledge of the gut microbiome. Are these bacteria, is this bacteria phylum the most abundant bacteria in the gut, or is it more the persistent sort of living?
It's the joint most abundant. So the Firmicutes and the Bacteroidetes, some people are dominated by Firmicutes, some by Bacteroides, but they're the 2 largest, almost dominant phyla, yes.
Okay, so let's see how many of these questions we get through. Is there an effective diet on which species that
we haven't done that analysis discreetly enough to give any kind of conclusion. We do certainly see some people are high and some people are low carriers. But I would point out as well that what we do in our assays is we deliberately spike every fecal sample with known amounts of a known phage. And then we can get absolute numbers. A lot of times people are expressing phage as relative abundance, the relative abundance of a phage to all the other phage present. But we found differences of up to 15 times in the actual total number of phage in different people's guts. And so you can have a situation where it looks like Crass phage are of low abundance in someone, but in fact they're people who carry very, very high numbers of phage. And so we need to look at absolute numbers as well. But we don't see any. We didn't with 10 people over 1 year, which is a very small group, we didn't see any seasonality, or we didn't see any obvious links to food, but then that wouldn't be a big enough number to pick that up.
Okay, let's move on to the next question, but you answered somebody else's question about season. Do you know whether all cells in a population are infected by the Crass phage, or whether it's only a subset of the population? And how does this tie in with the polysaccharide variability in Bacteroidetes cell populations?
Yeah, I think I saw that question come up. That's Leslie, I think. Yeah, that's a great question. It goes right to the heart of the work we're doing in the lab right now. And she's right on both sides. No, not all cells within a population are sensitive, and it is to do with polysaccharide variability. So the Bacteroides, at least intestinalis, in the presence of the Crass 001, switches between different surface molecules, and therefore it becomes sensitive and resistant. And actually, in the presence of phage, resistant cells will still become sensitive because it's just programmed into them to switch between different surface receptors. So there's almost, it's probably overplaying it, but there's almost a desire on the part of the bacterium to make sure the phage stays present as well.
Okay, so this sort of relates. Is there any idea, do you have any idea of what is responsible for the coexistence between the bacterium and the Crass phage? Is it related to resistance, low infectivity? I think you sort of said low infectivity, if I remember it.
Well, it's resistance, but it's a programmable resistance. The resistance switches on and off, and it varies between different receptors. So, you know, we use a kind of maybe a stupid phrase that the phage and the bacteria, they're not fighting, they're in a relationship. And that relationship sometimes involves fight. Some people lose and people get hurt. But in general, it's a positive relationship between the phage and the bacterium. And in evolutionary terms, that makes sense. I mean, it doesn't make sense for these things to coexist for such a long period of time if there isn't something in it for both of them.
Right. And do you know of any differences in infectivity between different Crass phage groups?
Certainly there are differences in that some of the Crass phage can't give rise to plaques. You know, we can't get them to plaque, and others plaque quite well. But that again is to do with the host as much as the individual phage. So there's certainly different levels of infectivity. If you do a classic burst size experiment, you see that the burst size is about 2 Crass phage per infected cell. But if you look through an EM, you can see hundreds of Crass phage within a single cell. So there's something very odd going on here that we're trying to unravel.
Okay, well, thank you very much, Colin. In the interests of time, I'm going to wrap up, finish the questions for you. And I would now like to thank everyone for being with us today to kick off this, this first virtual webinar series. Unfortunately, instead of the Viruses of Microbes meeting. And it was very nice to see that we had over 270 participants. So that's fantastic. The next webinar will be on the 15th of October at 4 p.m. GMT time. And that's going to be on virus structures and function. And you should visit the webinar website closer to the time for more details. And as you can see on your screen now, we'd like to encourage everybody to become members of the International Society of Viruses of Microbes, which is the society behind the meeting Viruses of Microbes and this webinar series. So visit the society website and become a member. A basic membership is free. So show your support for the types of activities that the society is trying to bring to everybody and to science and science communication on viruses of microbes. And I'd like to just thank everybody once again, and the organizers and all of the speakers and everybody who participated with all the questions. It's been a very stimulating first webinar, so that's fantastic. And look forward to seeing you all in the next webinar. I don't know whether the organizers have anything that they want to add before we finish.
We can just end up by thanking to the chairs, to the speakers. Thank you very much, and we will see you in the next iVoM.